Hi, everyone. Let's continue our discussion where we left off last time. On Monday, we were talking about the importance of immune memory and how the ability of our adaptive immune system to remember the sort of battle that we've fought against a novel pathogen um, and hold on to the ability to identify that pathogen much more quickly the next time we are infected by that same thing is a very critical part of how our immune system works. As I kind of described to you last time, always think about the fact that if your adaptive immune system had to be woken up in the first place, to get B cells selected and T cells selected and coordinate different types of effector cells, leading to the release of things like antibodies, that implies that whatever this pathogen is something that your innate system on its own, the way you were born with it, is incapable or not very effective in mounting a defense against. And importantly, I reminded you that during this lag in time, as your adaptive system is beginning to do things like select for lymphocytes, the pathogen is allowed in your body to essentially keep multiplying and you get really, really sick. I asked you to actually consider, and this is sort of no joke, even though it might, sort of on the surface sound kind of funny, the fact that when you are stuck at home in bed, drinking soup and watching TV because you're feeling miserable, you're dying, right? Because the pathogen is taking over, and the consequences, if this pathogen cannot be stopped, is that just like other pathogens, hey, they just do what they do, right? And they use up your body's resources and interfere with your body as an unfortunate side effect. And so even though you're feeling miserably sick, you always just figure that you'll get better after a few days, right? But you don't really think about the fact that your adaptive immune system has come to your rescue, and all the things that we've learned about with the selection of close enough fit immunoglobulins and the waking up of T cells and helper T cells, knowing what subtype of T cell to become in order to effectively coordinate a defense. All that stuff successfully happens, right? And the fact that you are sitting here today attending lecture is proof, right, that your adaptive immune system is very good at doing all those very complicated things, right? Thank goodness, they can do it even if you don't necessarily fully understand how they do it, right? But you're able to carry out those life-saving kind of events. So immune memory then, as I stressed last time, is important because just because you survived the first time that you were infected by a nasty virus, why would you want to have to suffer through that again, starting from square one the next time you're exposed to that same thing, right? Because maybe the subsequent times you're exposed to it, you're not going to be so lucky, right? And you might not survive, right? So immune memory is very important, and we evolved to have certain B cells and T cells and plasma cells stick around because they possess the sort of best kind of knowledge, if you want to put it that way, right, of what your body came up with in order to successfully beat the infection the first time. And so that if you are re-exposed to that pathogen again, days, weeks, months, years later, right, that is your starting point for mounting a defense. All right, so when we think about during an active infection, right, what is happening? So here we have what's going on with our T cells, what's going on with our B cells, right? So let's say during an active infection, you're going to have dendritic cells in like lymph nodes, for example, presenting evidence of, let's say, the virus, right? So let's say this is a virus particle here that your body's infected with. The dendritic cell presents the little red dot, which is sort of a peptide piece of the virus, and it selects for this particular naive T cell that happens to float in to the lymph node, on its way, going between the lymph and the blood. Right? As a result, because this is an active infection, the emphasis here is to save your life, right? You're not as concerned about, oh, can I have strong memory of this? Because that memory is not going to serve you any good if you die, right? So the emphasis is first and foremost in those T cells that are chosen to turn into effector T cells. Effector T cells, for example, if we're talking about helper T cells, then these T cells need to then start coordinating other immune cells to help fight the virus. If we are talking about cytotoxic T cells, right, then these capital E's mean that these are effector T cells that are now going to go out and in your body try to locate examples of cells in your body that are infected by this virus and are therefore presenting the little red dot on their MHC, right, on their MHC1. So as a result, very few of these sort of chosen T cells are actually encouraged at this point to become memory cells, right? The majority of them are activated as effector cells that go out and try to alleviate or discourage the spread of the virus. B cells, it's the same thing. When you have a naive B cell that binds to the virus due to its immunoglobulins, and then the B cell processes, chunks of the virus or whatever that it takes in, it presents the red dot to this helper T cell, right? And then this helper T cell will tell that B cell what to do, right? So that B cell will make many copies of itself. And most of those B cells, right, will stay sort of, well, the way the cartoon shows it, stays these yellow cells, because those will ultimately perhaps turn into plasma cells to release antibody, right? Because at this point in the active infection, you need to just focus on beating the infection, right? So very few of them at this point stick around as memory cells. But once the infection is waning, meaning once your body starts to beat the infection, then there's a shift. More of these T cells and B cells are encouraged to just become memory B cells or memory T cells to make sure that you have good, strong immune memory of this. Now that it looks like you're in the clear, right, and you're not going to be killed by it, killed by this virus this round. So memory cells are crucial for a strong secondary response. So a full-blown secondary immune response occurs when pathogen reinfects, right? You're introduced to this pathogen a second time, and the memory B cells and memory T cells go to work right away, right? Because you don't have to start from scratch and select for new ones, right? You have the ones that are sticking around. This is particularly the case if in reality, this secondary infection still ends up making you kind of sick. That's even more emphasis on your immune system really paying attention and realizing, hey, this wasn't just a one-time deal that happened like 6 months ago or 6 years ago. We are encountering the same pathogen again, right? So our secondary full-blown immune response is really huge compared to the primary response. So that's mediated by our memory cells. So some advantages of having memory cells already present are, for example, in this case, here's the googly eye drooly virus, right? That's trying to reinfect. So this is after many months or years after the first time that you got sick from this virus, you have these memory B cells, right? These memory B cells that have the IG that right away can bind to certain surface features, certain antigens, and the B cell can turn into a plasma cell and make lots of high quality antibody right away, right? Enough to be able to really help out in neutralizing the virus, right? So as part of your memory, that means that the number of these antigen specific memory cells that you have compared to the by chance brand new naive B cells that are coming out of your bone marrow that day, right? You should have more memory cells that recognize the virus than the brand new naive B cells, right? So they're outnumbered like 100 to 1, right? So you have more of these cells that are specific right from the start. The activation of these memory cells is much faster. So we said that normally during a primary infection, the first time when your body is learning how to deal with this virus, right, it might take more than a week before you really see plasma cells releasing lots of antibody, right? And during that week, you are getting super sick. Right? But in a secondary response, right, because you're starting with these memory cells, it's much faster. Within maybe just 4 days of reinfection, you start seeing your antibody titer shoot up, right? And 4 days is pretty fast because remember, right, when this virus reinfects you again, your adaptive system, sure, it's ready in the sense that there's memory of it, but really your innate system has first crack at it again, right? So your complement proteins, macrophages, they try to go ahead and neutralize this pathogen, right? Just like they did the first time, right? So after a couple days, a few days, when it's pretty clear, right, that this pathogen has come back, right, you might see then the plasma cells really beginning to up the amount of antibody. Right? Memory B cells express higher levels of certain proteins on their surface that are meant to increase the ability of communication and alert your immune system that the googly-eyed virus has come back. So for example, memory B cells express higher levels of MHC2 on their surface, right? And also co-stimulatory molecules like the CD40 and the CD28, so that these cells, right, can communicate with other immune cells a lot more readily and a lot more quickly to make sure that these memory B cells activate and sound the alarm. And also importantly, memory B cells have already undergone the class switching between different types of IGs. Remember, all naive B cells start off just making IgM on their surface. Right? But during the course of an infection with like this googly eye drooly virus, right, the IgM is not going to be what saves you, right? It's going to be high quality alternate versions of IGs like IgG or maybe IgA, for example, right? And so the memory B cells that are sticking around have already done that class switching so that when the virus comes back, they already are making the form of antibody that is most effective in dealing with this particular type of pathogen. Right? So repeated exposure then to a pathogen, right, boosts your overall antibody levels, and it provides opportunities for your B cells to still continue this whole hypermutation process of trying to get stronger and stronger antigen binding affinity, right? Because remember, at the end of the first time you were sick, your first exposure, you came up with really good quality IGs, right? Good enough that that finally was good enough to neutralize and beat the pathogen. But that doesn't mean that those IGs necessarily are the best possible ones that you can come up with. It's just you stopped there. You stopped there because that was the end of the infection, right? So whatever cookie recipe these hypermutated, B cells came up with during your primary infection, that's what you have, right, as your memory. If you are exposed again to the same pathogen, right, then those memory B cells can also serve as the starting point to continue with the hypermutation. So let's look at this figure here and think about this. If you are infected by something one time, there's a reasonable chance that whatever this thing is in your environment, right? So there's a good chance that you'll be exposed to it again. But until you are exposed to it again, your immune system, it doesn't quite know how much effort or resources it should devote to making a strong immune memory. Right? I think I pointed out to you before, if our immune memory, if part of that is things like the presence of memory cells, B cells, T cells, and the presence of elevated antibody levels floating on our blood, it's just not simply practical for your body at this moment to have in it high levels of every single memory B cell, T cell, and every single antibody that your body made in the course of your 20 plus years of fighting pathogens. Right? There's just not enough physical space in your body. Right? So as a result, even though a primary infection gets your adaptive system working, and hopefully there's some memory of what your immune system came up with, right? Your body doesn't really know if you're going to encounter this pathogen again, a week, a month, 10 years, or maybe never again, right? So following that first infection, Your memory is good, but it could get a lot better. If you're infected again by that pathogen, that's when your immune system says, whoa, hang on here, right? We do need to really pay attention to this because this is back, right? The googly-eyed drooly virus is back. So it's really worth it for us to invest in making very strong immune memory. So if you look at this here we are showing changes in the body in terms of levels of antibody, as well as, levels of antibodies at the top, antibody amount, as well as how good these antibodies are, right? The quality of the antigen binding domains of these antibodies. And I think it's very timely here that instead of using examples of infection, what are they using? They're using examples of immunization, right? Of getting a vaccine shot. So you can see following the first shot of a vaccine, of course, your adaptive immune system is encouraged to go ahead and try to select B cells, T cells, do hypermutation to come up with good antibodies in response to it, right? And the whole premise or the whole concept of a vaccine is you are presenting to your immune system those particular antigen features, the three-dimensional shapes that are determined to be the good shapes for your body to use as the basis of antibody selection, right? But you're presenting it in a way that it's not the intact live virus, right? Because look, Mother Nature intended it to be, you have to get the real virus and get sick from it, and it's up to you to survive, right? And beat it. And that's how you get your immune memory. But vaccines, right, they present the antigens that you want your immune system to learn from without presenting the actual threat of the pathogen itself, right? So as a result, your adaptive immune system has time to build up and select for lymphocytes, and you're not actually, at the risk of dying from it. So as a result, following the first shot, you can see the quality of IGs go up, right? So this is due to hypermutation, getting better and better fit IGs. And you can see the levels of antibody circulating around in your body, like IgGs through your blood, right? It goes up after about 2 weeks, right? So see this peak here, right? It corresponds to 2 weeks after the first primary shot, right? Because that's when it'll peak. And then it begins to taper off. Why? Well, because there really is, not a huge threat here, right? The vaccine has probably by now been neutralized by your immune system. And so your immune system just says, okay, at this point, all right, that's fair enough, right? We developed some antibodies against it, and we're going to keep some antibodies around, whatever. That's why after you get the second shot, right, this is when you really make your immune system pay attention and say, hey, it's not good enough that you mounted and chose some antibodies the first time, right? We really need you to take this seriously. So following the second shot, you have a much stronger immune reaction, because that's how Mother Nature designed us, right? Because the fact that you're exposed to this thing a second time means, right? It's in our environment. We do need to develop really strong protection against it. So as a result, look, your antibody amount shoots up dramatically, right, after the second one. And then the quality of your IgGs continue to improve, because the memory cells that you developed after the first shot, right, form the basis of further hypermutation to get better and better and better. Right? That is why you've probably heard in the news, maybe you yourself have experienced this or through a family member, right, that most people don't really have a strong reaction to the first COVID vaccine, the first shot, right? So you get the first shot and then you wait several weeks. I think typically it's like 3 to 4 weeks, right? So that this primary immune response can take place. And then you get the second shot to really make your immune system take this seriously. And that's why when there are bad side effects, it typically happens after the second shot, right? Because then your immune system really goes overboard, and many of the side effects that are associated with your immune system really waking up. Things like fatigue, things like, fever responses, things like drowsiness and, aches and things like that. That's all part of inflammation, right? That your immune system is using to kind of wake things up. And that's why the second COVID shot is the one that typically makes people feel miserable. So, in other words, if you feel bad after your second shot, at least you can take some comfort in knowing, wow, that's because my immune system is really taking this seriously, right? And hopefully that means your immune system is really doing its job and building up a strong, long-lasting memory. If you don't have a set of side effects from the second shot, don't worry. It doesn't mean that your immune system isn't paying attention, right? It just means that in your particular body, in your situation, right, your immune system is not eliciting the same types of strong side effects. But, it surely is still building up strong immune memory. Right. That's also why, right, there are some reported cases of people who react strongly after their first shot. And it might very well be that those are people who previously were exposed to COVID and didn't even know it, right? Because not everybody develops symptoms of illness from exposure to COVID. I think you know that, right? Some people can just kind of carry it and pass it along. But when they get their first COVID shot, right, that is essentially functioning as the second COVID shot in other people, right? So they end up having their big secondary immune response just after their first shot. That's also the reason why the CDC recommends that if you are currently recovering from a bout of COVID, right, that it's still good for you to get the vaccine, right? Because you've had your natural primary response to it, right? And now you might as well benefit from using the vaccine as your secondary response to really boost your system. And that's also why they say if you're recovering from a bout of COVID, you should wait. You should wait a few weeks before you get your shot. And we will actually talk about that a little bit more in one of the subsequent slides here. All right. So I wanted to stress with you, with each exposure, activated B cells undergo somatic hypermutation and improve their Ig affinity, right? So your immune system is constantly working towards better detection of the pathogen that made you sick many years ago. So there most definitely are cases or instances in your own life where you were exposed to something as a kid, right? And it made you viciously sick, like you were throwing up and had diarrhea and all this stuff, right? But then you beat it. Obviously you beat it, because otherwise you wouldn't be in college attending this class now. But just think about this. That might have happened something like, 15, 20 years ago, right? In the last 15, 20 years, you probably were exposed to that same thing again and again and again, right? Over the course of the last couple of decades. Each time you didn't realize you were exposed to it again because you had strong enough immune memory that you could neutralize the pathogen before it had a chance to make you sick, right? But in the background, your immune system used each opportunity as another reminder that it should be building stronger and stronger and stronger antibodies against it, right? So the antibodies against whatever this, let's say, cold or flu was that you had when you were 6 years old, the version of the antibodies in your body now are way better than the version that you came up with as a kid those many years ago when you needed to come up with it to save your life. Right? This is an interesting thing to think about. All right. During a secondary infection, let's say the googly-eyed virus comes back again, or, let's say it's COVID, right? So you're immunized or you experienced a primary infection with COVID, you beat it, right? So now you have immune memory, okay? And then all of a sudden, right? You're exposed to the virus again, right? Think about this. In that type of scenario, you are depending upon the fact that you have elevated levels of memory B cells already present, right? And those are the ones that you want to rely upon to actually mount the secondary defense, because they all have really good quality cookie recipes, right, for their IGs. And the whole point is, you don't want to waste time having to go through the selection of brand new B cells from scratch. But right? Every day, you have hundreds of millions of brand new B cells coming out of your bone marrow. And certainly there's going to be a population of those that are the close enough fit puzzle pieces to the antigen of the virus, right? So those will also start to want to bind, right? But that just gets in the way, right? Because why do you want to worry about reselecting and starting from scratch these naive B cells when in reality you have these great memory B cells to work with? So our body has a way to actually discourage naive B cells during a secondary immune response, right, from being chosen or selected. So let's look at this. During our primary response, the first time you're exposed to COVID or the flu or whatever, right? Here you have a naive B cell. It has close enough fit IgM on its surface. You can see, right? It's pretty lousy fit, but it's close enough, right? And so this B cell then is activated, and through all those processes that we talked about, it might become an antibody producing plasma cell, right? And the first thing it's going to do is produce some low affinity quality IgMs as antibodies that are floating away, as other brother and sister B cells, because this thing's going to divide and make many copies of itself. Remember, some of them are off in the germinal centers of the lymph nodes doing the hypermutation. So as a result of those other B cells doing the hypermutation, you end up beating this infection because you make high quality antibodies, right? Maybe IgGs now, right? That represent much better fit to the antigen. Right? Now, let's say months or years later, you are exposed to the same pathogen again. Right? In this case, you're going to have memory B cells that are sticking around, right? But you also, on that given day, happen to have a pool of brand new naive B cells that just came out of your bone marrow, right? Just like that very first day many years ago when you had to select for them. But now you don't want to bother selecting for them, right? Because that's going back to square one. Look, if this naive B cell is floating around, right, with close enough fit IgM, it's like, hey, check it out, right? I have close enough fit, Ig, right? So you might have these B cells, these naive B cells, kind of interfering with the ability of the memory B cells to properly bind and detect the virus, right? So this is something that's interesting. This is a key thing that happens. Because this is a secondary response, you already have high quality, these orange antibodies made, right, as part of your memory from your primary response. Naive B cells have on their surface Fc gamma receptor, Fc gamma R2B1. This is green thing. This Fc gamma receptor can bind to the stems of IgG, right? That they bind to the gamma Fc region of antibodies. So you can see this naive B cell that came out this day, right? It's actually able to coat itself with these high quality antibodies that are floating around, right, as part of your immune memory. If one of those binds to the antigen, and you imagine it's going to bind much stronger than the crummy yellow IgM over here, that's part of what this B cell knows how to make, right? It will actually send this orange antibody binding will send a signal through the Fc gamma receptor to this B cell that tells it, hey, you know what? That's great that your IgM is close enough fit, but we actually don't need you because we already have high quality IgGs, these orange IgGs floating around. That means we've got this covered because we already have a pool of B cells that are memory B cells that know how to make this orange IgG. So as a result, these naive B cells that bind antigen during a secondary response are actually discouraged from activating so that they don't mess up or get in the way of your good quality memory B cells doing their job. So as a result, the good quality memory B cells that have, right, the know-how of making this high quality IgG, right, they're the ones that get preferential access to bind the pathogen. And so that's how you then can wake up your immune memory much faster. So there are certain instances when we can actually use this therapeutically. So one of the complications that happen during pregnancy is when the mother's blood type is different than the baby's blood type that she is carrying in the womb. As we said, mother's blood and baby's blood don't normally mix. So blood cells and things are kept separated by the placental barrier. It's only things like nutrients, gases, waste products, things like that can diffuse back and forth between mother and baby. However, things like IgG proteins, like antibodies, can go through the barrier. They have to be actively transported across. It's not like these antibodies will just float through cell membranes, right? Because they're proteins. Proteins don't typically float through cell membranes. So they can be taken across and put into baby. And that's a critical way, as we learned, that mom can equip her unborn child with a big library of IgGs to help protect baby when baby is born and baby's own immune system hasn't had a chance to fully activate yet, right? But if you have a situation, for example, during delivery, delivery is a very messy, kind of bloody situation. You're going to have some mixing of baby's blood and mom's blood, right? So as a result, you may actually end up with mom producing antibodies against baby's blood type, right? So the E here just means it's an erythrocyte, right? So it's a baby's blood cell, and maybe baby is Rh positive, let's say. Okay, and mom is Rh negative. So there's actually a possibility that mom has naive B cells that have Igs that bind the Rh signal, right? And then mom might actually develop during this following birth, right? Her immune system, even though baby's born, mom's immune system might actually be like in the background going like, oh, we should select and build up some memory against this weird Rh factor, right? As though your immune system really knows what it's called, right? But it'll build up immune memory against it. And so what might happen is, unfortunately, when mom gets pregnant again, and the second child, in utero is also Rh positive, the baby is Rh positive because of dad, right? Dad is contributing the allele for the Rh. The problem is mom has immune memory against baby's Rh positive status, right? And the problem is those antibodies, those IgGs that are made by mom's immune system, right, are transferred just like any other IgG into baby's body, right, while baby's still in the womb. And as a result, those antibodies might actually start targeting and destroying baby's own blood cells in the circulation. So the way we prevent this from happening, so this results in the baby born anemic, right? Meaning lower levels of red blood cells than needed to properly carry oxygen and things like that, right? Because ironically, mom's antibodies that are part of baby that's meant to protect baby are improperly destroying baby's own blood cells, right? So the way we can do this is during pregnancy, when it is determined that this situation is going to happen, like mom is Rh negative and the baby is Rh positive, mom can actually receive an infusion of anti-RHD IgGs, right? So the IgGs are intentionally put into mom so that any naive B cells that are coming out of mom's blood, bone marrow, right, if they start binding to the evidence of baby's blood type in there, they're discouraged from actually activating, right? Because these IgGs are present and they discourage mom from mounting a defense, right? So this would typically happen, I think they give it pretty close to birth. So the idea is these antibodies are not transferred into baby. They're really part of mom, meant to kind of discourage mom's body from actually thinking that the exposure to baby's blood means that it has to learn and select for antibodies against it. In terms of T cells, we have two populations of T cells. We have what are called central memory T cells that stick around, right? And these are primarily helper T cells. So imagine you have this memory helper T cell. So it's a CD4 positive helper T cell that's sticking around as a memory cell. It has the memory of the googly-eyed virus, right? When the googly-eyed virus comes back again, right, this memory T cell is the one that can quickly help encourage memory B cells that also remember the virus to quickly activate. Then we also have effector memory cells. These would be more like the CD8 positive cytotoxic memory cells, right? So when the virus comes back, these memory T cells, boy, they recognize the virus right away because this guy's bumping into this cell in your body, and this cell is doing what? It's presenting evidence that it's infected by googly eye drooly virus, right? So very quickly, these memory T cells go to work and tell this particular infected cell to undergo apoptosis and die, right? Hopefully discouraging the spread of more virus particles. All right, let's get to the good stuff, the stuff that I know you're interested in hearing about, and that has to do with COVID, right? And what the deal is with COVID. Right? So COVID, of course, is an example of a global pandemic. And as we discussed before, pandemics are often caused by certain viruses that have the right combination of being highly infectious. They are very detrimental and very life-threatening, but at the same time, right, they are not super fatal. Because I know this sounds kind of weird if you think about it, but if the COVID virus were super fatal and let's say killed everyone that it infected within a week, then that itself would sort of limit the spread of the virus, right? I know that's not a nice way of thinking about it, but it's kind of true. It's almost like the virus would just sort of burn itself out, at the price of killing everybody very quickly, right? But so something like COVID has the unfortunate combination of being highly infectious, so it is transmitted very easily, and it's not super deadly, but it's bad enough to make you potentially really sick, right? So that's kind of like the worst combination. So how does something like that happen, right? I mean, where does COVID come from? Out of nowhere, right? A lot of times, viruses like this are recombinant viruses. That means they represent genomes of different viruses that have come together, by chance to form sort of like a new version of a virus. For example, let's think about it this way. Let us imagine that there is a virus. Let's say this is like a human, I don't know, like a flu virus or something, right? So it's very good at infecting people cells. It's very good at infecting people cells, but fortunately, it's not super deadly, right? I mean, look, let's face it. Yes, I know every year, tens of thousands, if not hundreds of thousands of people around the world do die of the flu, right? But I think you all know that if you get the flu, right, you're not really worried about dying, right? You're going to be sick and get better because the flu itself is not super fatal, right? So here is the human version of that flu, right? So that's the version that makes Homer really sick, right? And let's say there's also another virus, a virus that infects birds, right? Similar thing though. This version infects birds and it makes the birds, pretty sick, but it doesn't kill them super fast, right? But the point is that this bird virus, right, isn't the right shape to be able to infect human cells. I know this is kind of weird if you think about it, but you go walking along the sidewalk and you're like, there's a whole flock of pigeons that you're like, get away, right? You might be breathing in bird viruses, right, from those pigeons. But the fortunate thing is those viruses, they don't have the ability to enter your cells, right? So as a result, they just are naturally over time destroyed by your immune system, okay? But let's say you have a pig whose surfaces of its cells, right, have certain structural features like glycoproteins, things like that, represented by the little purple lollipop here, right, that are actually suitable for the human version of the virus to infect it, and also the bird version of the virus can infect it as well. Right? Now, I know you think that's weird. You're like, what? Can it really be that multiple viruses infect the same cell at the same time? Wouldn't it actually be more surprising if that wasn't true, right? Because there's no weird unwritten virus rule that says like if a virus is infecting a cell, then other viruses that are being breathed in, even if they're a different type of virus, are not allowed to infect the same cell, right? In fact, one cell might be infected with thousands of particles of two different types of viruses that unfortunately are being taken in. So that's what happens in this pig cell, right? Both the human and the bird version of this virus infected. So as a result, all that genetic material is getting mixed up inside of the pig cell. Right? Yeah, the pig gets sick, right? But importantly, the genomes of these two different viruses gets mixed up. And so what do you end up with? You end up with a recombinant version of this virus that is unlike either of the previous ones. And unfortunately, right, maybe it is the combination that allows it to infect human cells, but unfortunately, it carries viral elements, viral genes that the human species has never encountered before, right? Because these were all things that birds had to deal with. So what does that mean? It could very well mean that our immune system, right, is very poorly equipped to quickly deal with this. And this virus might spread very quickly and rapidly in our bodies, right? So that's why things like COVID, right, are particularly dangerous. So coronaviruses, so COVID is an example of a coronavirus. So it's an RNA virus. It carries its genetic material around as RNA, right, not DNA. There are many examples of coronaviruses like COVID making the jump from other species into people, right? So this goes back all the way to, let's say, the 1100s, right? There's evidence of viruses jumping from bats into people, or SARS, right, in 2003, jumping from bats to other intermediate mammals into people. MERS went from bats to camels to people. We don't know at this point what the originating animal is where the COVID-19 virus came from, the SARS-CoV-2, right? I mean, early on, there was lots of speculation that it could be bats, it could be pangolins, things that are, sold in meat markets. But the truth is, even now, we still don't know what it is. But that's what has happened, right? By jumping through different organisms, the genomes have gotten mixed up so that they have the combination of being able to infect our cells, but yet presenting viral components that the human body is not used to seeing, right? So it's the first time in the human species that we've had to deal with these particular types of things in the virus. So here's the COVID-19 virus, the SARS-CoV-2. And so it's very closely related to previous SARS viruses, right, like SARS or MERS. And so SARS stands for severe acute respiratory syndrome, right? So we now call this SARS virus 2, right? And so really it's just genetic material, RNA, that has some proteins in there, proteins that take care of maintaining the RNA and also replicating the RNA, surrounded by a capsule, which is a lipid capsule, very much similar to the cell walls of our cells. And it's studded with these protein spikes. So these protein spikes are the right shape to bind to certain proteins that are on the surfaces of our cells, for example, the cells of our respiratory tract. In particular, this one receptor known as ACE2, which is an angiotensin converting enzyme that's normally used and received by the cells of our respiratory tract as a way to modify things like the constriction of blood vessels and also the tightening of smooth muscles, right? So it just so happens that this is how the virus gains access into our cells. So these glycoprotein spikes, as with most viruses have these types of structures on the outside that represent the three-dimensional shapes that bind to the host cell and get the virus in. These serve as your best sort of potential antigen signals that your immune system is going to actually choose antibodies against, right? Because they're positioned on the outside of the virus by design, right? And they have distinct three-dimensional features that make this virus unique because they have 3D features that bind to this ACE2 receptor. So here's the protein spike. It's a glycoprotein, so you have certain sugar groups and other polypeptide chains in here, right? So this is the antigen that's really the basis of our COVID-19 vaccines. So as we discussed before, in a very basic kind of sense, the idea is if you can introduce just this antigen, right, without it being part of a virus particle, if you can introduce that into a person's immune system, then you're giving the person's immune system a sneak peek, right? You're giving it a heads up that this is a foreign shape that you want the immune system to go ahead and start selecting antibodies for, so that the person's immune system can build up immune memory without any risk of them actually getting sick at all, because this is not the intact virus. This is just a piece of it, right? The piece that matters in terms of your immune system recognizing it, right? So you have B cells that have IgGs that recognize the antigen. You will also have helper T cells that have T cell receptor that recognize peptides from the antigen, right? And then those helper T cells will work with those B cells and encourage those B cells to do things like turn into plasma cells to release Ig antibodies. Some of these B cells will also be encouraged to go into your lymph nodes. Oh, no, they're already in your lymph nodes. They'll encourage to go to the germinal centers of your lymph nodes and do hypermutation, right? So over the course of a couple weeks, even though you're not sick at all, because this is just the antigen from the virus, not the virus itself, hey, your immune system treats it as though this is really a threat, right? And so you mount an immune defense and you build up good quality memory B cells, T cells, and they may actually also class switch, right? So because this is maybe a viral protein type of thing, you might actually produce things like IgAs, so memory cells that produce IgAs that can then go ahead and start lining your respiratory tract in defense of anticipating the virus, right? So here are some other views of that glycoprotein spike. You guys don't need to know this for the exam, right? Because this is more advanced stuff for the exam, but I think you like seeing this, right? Because what it allows you to see, if this is the glycoprotein spike, this is the antigen, but on that antigen, because it has unique three-dimensional landscape, many of these surfaces on it represent different epitopes, right? Or different shapes that your antibodies can bind to. And so you'll select for, a population of many different naive B cells, each that have close enough fit to a different one of these epitopes. And all of those B cells are the ones that are going to be chosen and selected as part of developing your immune memory. Right? So some challenges. The free-floating antigens, so if you just had the glycoprotein spike in a syringe and you injected that into your body, that's not really optimal as a vaccine. Why? I mean, it's good because there's no risk of you getting infected, right? You're not going to get sick from this. But the problem is your immune system's not going to pay attention to it, right? Because it's just another random protein that's easily lost in the whole mix of all kinds of proteins that are floating through your blood, right? Or even in your muscle tissue where the shot is given, right? Your immune system is not going to really pay attention to that. So one way to get things to pay attention is if you are going to use the antigen directly as the basis of your vaccine, you need to have other ingredients in the vaccine formulation to really get your immune system kind of alerted. And those other ingredients are what we call adjuvants. So certain types of oils, certain types of lipopolysaccharides that are actually derived from bacterial cell walls, certain types of metals like aluminum, right? If they're all part of what's injected into you, those substances, right, we now know really help induce inflammation and they get your immune system to respond, right? But unfortunately, those adjuvants are also associated with certain types of bad side effects like fever, potential shock, for example. Either way, this is not the natural mode that your immune cells would have actually learned to pay attention to something, right? Because there's no real virus there. There's no real infection going on. Therefore, the immune memory based upon this might not be that strong. Right? We also learned that sometimes you can use attenuated whole viruses, viruses that have been altered so that they're lazy, right? The lazy virus. These are good as the basis of vaccines because they actually do infect your cells, but they're just so lazy and slow that your immune system has plenty of time, right, to select your B cells, T cells, and neutralize them before they actually make you sick. But as we said, the downside of that is because it's actually intact virus, there's also the possibility that it might mutate inside your cells, in which case, oh man, you actually ended up catching COVID-19 just from the vaccine itself. That's bad, right? So that's why these new types of vaccines that are coming out, like the Moderna and Pfizer vaccines, right, are exciting because they're based on mRNA, right? What's mRNA? Remember, when you have a gene inside of your cells, whether it's your own gene, right, as part of your genome, or if it's genes that are brought in by a virus particle, right, those genes, right, in order to be turned into protein, right, they have to be first transcribed and processed as mRNA. and then the mRNA is translated by your cells to create different proteins, right? In the case of coronaviruses, they carry RNA in the first place, right? So for example, the RNA that codes for the glycoprotein spike part of the virus, right? It comes in as part of the virus genome. So if you went ahead and actually designed a vaccine that is just the mRNA for that particular glycoprotein spike, right, not the other parts of the vaccine, because you don't want your cells to make those, and you put that in your cells, then your cell will make the glycoprotein spike as though it's being infected, right? And it will present the glycoprotein spike to your immune system in a way that closely mimics an actual infection. Right? But again, because there's no real infection, because it's just the mRNA that makes your cell make the glycoprotein spike, right? There's no risk of you actually getting sick. So these mRNAs are what we call modified mRNAs or mod mRNAs. So this is what the Pfizer and the Moderna vaccines. So they're engineered, right? So these mRNAs have 5' and 3' caps as part of the mRNA sequence that make it really look more like a normal mRNA that our own cells would make, because after all, our ribosomes are geared up to do a better job of making those types of mRNAs translated into proteins, right? They also have 5' untranslated regions in the mRNA that really encourage the ribosomes to take the mRNA, bring them to the endoplasmic reticulum, and you have higher efficiency of making the glycoprotein spike, translated into the endoplasmic reticulum. And importantly, they also have substitutions. Different bases or nucleotides in this mRNA are altered. So instead of uracil, right, or instead of cytosines, they actually engineer this vaccine version to have modified bases, pseudouridine and 5-methylcytosine. Why is that? Because by having those types of signals that are very foreign to your cell, right, those don't impact your ability of your cells to translate and make the protein, right? So they don't actually negatively get in the way of your cells making the glycoprotein spike, but they do get in the way of your cell's own pathogen detecting machinery to detect that there's viral mRNA present. Okay, because remember we learned as part of a viral interferon response, certain types of foreign, like double-stranded RNAs, things like that, are detected by our mitochondria, right? There are different proteins as part of our immune system that recognize foreign genetic material, and that's what mounts the interferon antivirus defenses. Right? In this case, if you modify the RNAs, those defense mechanisms don't recognize it, so they don't get in the way of your cell making the glycoprotein spike. Okay, last slide. Right. So this is typically then what happens. Normally, the COVID-19, the SARS-CoV-2, it binds to the surface of your cell by having these light blue glycoprotein spikes that bind to the ACE2 receptors on the surface. That's what gains entry, and the virus releases its RNA. Right. So certain proteins that are also released by the virus that it was carrying around copy the RNA and start making many copies of the viral genome, right? And then at the same time, this RNA, it doesn't have to go through the nucleus at all because it's already RNA. And so your poor ribosomes in your cells begin to make the different viral components. For example, stuff that makes the envelope proteins, stuff that makes the membrane, and stuff that makes the glycoprotein spike, right? So as a result, those proteins are all made in the endoplasmic reticulum, and they get assembled, right? They get assembled inside of the cell, going through the Golgi into new virus particles that then are released to infect other cells. So what we're saying is, forget about all these other mRNAs, right, that are part of what the viral genome carries. If you just introduce the one that you care about, that you want your cells of your body to make as part of the vaccination, that's the spike protein one, right? Then your cells will just make the glycoprotein spike and have that on its surface, even though it's not going to make the rest of the virus, right? And therefore, you get a very strong immune response, right? Because this looks like your cell is infected with virus particles, right? And your T cells, B cells, they're used to seeing antigenic signals presented on the surfaces of your body's cells. And that's how you mount a very strong immune response, because this is what Mother Nature, sort of designed our immune system to recognize. Okay. All right. Okay. I'm going to stop there because I know some of you have to get off to lab.